Hi, my name is Paul, and this is my video explaining my what I understood about what I've been reading about Imatinib so far. I think this video right now is going to be a little broad because breaking into the subject was a little bit there was a lot of new information I had to learn. So and the new information was like really easy to condense. So I'm just gonna explain this. It's gonna be like maybe 10 minutes, and then my next videos will be a little bit more in depth because my inquiries are a little bit more specific. But here is my understanding of imatinib. So let's first start with like the content. So imatinib is a targeted therapy used for CML, which is chronic myelogenous leukemia. It's leukemia that starts in the myeloid cells in the bone marrow. And then I'm going to go on to what imatinib does in a more broad sense, and then further inquiries. So first of all, CML is chronic myelogenous leukemia. It's a slow-moving leukemia that starts in myeloid cells. And myeloid cells are essentially cells that are can turn into one of three things. And these three things are part of something called the myeloid lineage. These three things are platelets, white blood cells, and red blood cells. But for CML, this is primarily affecting the white blood cell count in the cells. And then eventually, like all leukemias, the uh CML causes an overabundance of myeloid cells, which leads to very deadly symptom. Now let's get into how does CML form. So CML is formed by the presence of something called a Philadelphia chromosome. A Philadelphia chromosome is so a human body has 23 pairs of chromosomes per cell. This also includes myeloid cells. And these chromosomes are different shapes, different sizes, and they contain different genetic information. The Philadelphia chromosome is a mutation that was discovered in Philadelphia, hence the name, and it's a translocation of chromosome 9 and 22. What this means is, but you could kind of tell based on the visual, but what this means is some of the a section of chromosome 22 and a section of chromosome 9 swap places. So what this does is you get this extra short chromosome 22 and this extra long chromosome 9. This extra short chromosome 2 is what's called the Philadelphia chromosome. And this is so important because I prefaced this earlier. Chromosomes contain different genetic information, right? Each pair of chromosomes contain different genetic information. And they are that way because the cell reads these genes and says, hey, we're going to code these proteins based on the genes that are in the chromosomes, right? But then once this translocation occurs, this BCR gene and ABL gene, which both code for proteins individually, two separate proteins individually are what these genes code for, end up right next to each other. And the cell reads this and codes for one big protein called the BCR ABL tyrosine kinase, which is which is a tyrosine kinase. So a kinase is essentially an on switch. What it does is it transfers a phosphate from ATP onto other proteins and and oftentimes starts a signaling cascade. And the tyrosine kinase in specific transfers the phosphate onto the tyrosine residue on the protein. So what this kinase does in specific is well, first of all, the BCR-ABL gene, which is the fusion, which is what happens after in the Philadelphia chromosome, it is called an oncogene, meaning it causes cancer. It's the gene that causes the leukemia. So what happens is in CML, this oncogene codes for this BCR-ABL tyrosine kinase, which is just an on switch, right? And what happens is the protein opens this activation loop to receive ATP, right? So, and because it's a leukemia, it's constitutively on, meaning that the protein is almost always in the open loop formation. Okay. This is where imatinib comes in. Okay. Imatinib is essentially a drug that binds to the closed loop of the protein. So, obviously, based on what I just said, that's not very often that the loop for the protein is closed. That means there's very small windows of time for imatinib to bind. And to explain what exactly binding to the closed loop means, it basically now the protein cannot open the loop because imatinib is there. It cannot open the loop because it can never open the loop. The signaling cascade that starts because of the transfer of the phosphate from ATP cannot happen because ATP is not there to bind because ATP can never bind to the open loop because the open loop is specific or organizes itself efficiently to fit ATP. So if it can't do that, then the ATP never binds, and then the CML never starts. Now, for further inquiries based on this, I know this was like a little bit of a shorter video compared to like how information dense this topic is, which is why I'm going to get into it a little bit more in the further videos. So, firstly, my next video is probably going to be on a little bit more on the chemistry specifics of imatinib. I think that's really interesting. I also plan a lot of these papers that I read, they do talk about mutations in CML. And I'm like I still don't know I'm very new to the subject. I still don't know what that means. So I am also going to be looking into that in the future. So that's probably what's my next video is going to be about. And then here are my citations. So my first video, which gave me my background, my first article that gave me my background on CML is this chronic myelogenous leukemia article found on Mayo Clinic. My second paper that gave me the information on the signaling cascade was this BCR-ABL1 tyrosine kinase complex signaling transduction challenges to overcome resistance in chronic myeloid leukemia. And this has first authors Gustavo P. Amarante Mendez and Amir Rana. The third article which helped me, which was this BCR-ABL1 fusion gene from cancer.gov. I was just a little bit confused on exactly what it meant for these genes to be fused, what this translocation meant. This was a very helpful article in explaining that. And then this is a review article, Imatinib, Breakthrough of Targeted Therapy, with first author Nida Iqbal and second author Naveed Iqbal. Thank you, and I'll see you in the next video.